the Macula Vision Research Foundation, and he got together a group of scientists about 1997 and said to us he wanted to fund research in this area. What should we fund research on? And I can tell you we didn't have a clue because so little was known about the disease. And the real breakthroughs have come from genetics. Because of the Human Genome Project, you can now look at all the genes in a person and compare all the genes and the variants of them in a normal person without macular degeneration to populations of people with macular degeneration. And when you did that, you found that that was actually, macular degeneration was the first disease where looking at the whole genome had a home run. And the home run was the discovery that a particular protein called complement, which is involved in controlling inflammation, there were different variants of it than in people, in wild-type people, in people without macular degeneration compared to affected individuals. And so the insight there was that part of this disease process is due to unregulated inflammation. So in a person without the particular genetic predisposition, that if they had a little minor insult to the eye, let's say a little blood clot, or maybe because they smoked or whatever, there might be a little inflammatory event, but the body would tone it down, and nothing would happen. Whereas if you had the variants of these genes involved in inflammation that were less capable of dampening the inflammation process, that would initiate the macular degeneration process. So that was a great insight, and it hasn't led to treatments yet, but I think in the long term it will. And so similar research has now led to a whole bunch of genes that predispose you to getting this disease. And in the long run, that'll have clinical outcomes, but that'll take a while. There's always a huge gap between, which politicians don't understand, between the investment in the basic research that leads to discoveries like this and benefits to people, which generally take 20 or 30 or 40 years to achieve.
That's right up the questions I wanted to ask now is, what is the process that goes from your work in the research in the laboratories to the clinical applications for us that are needing to be treated, and some of the obstacles you come across when you're going from laboratory research to clinical applications, just for us lay people to understand?
That's a fabulous and important question, and this is a really exciting. The discovery of the genome means you can have in a dish, you can have a working version of any of the 23,000 proteins. Okay?
Yes.
So let's say you find protein 6 is involved in a particular disease. And if you want to dampen, you want to increase or decrease the activity of protein 6. So what you can do now is take a library, it's called, a bank essentially of 200,000 chemicals that you may not have any relationship presently to medicine, and you pass them through and see if they bind to protein 6. And then if they do, then you see, okay, do they increase or decrease the activity of protein 6? So this is changing the way the pharma industry is working in a dramatic way, because this is, without going into the current or the historical way of finding a drug, this is a new way of finding drugs. And it's going to yield massive returns, but it's slow and difficult. So let's say you find a molecule, something that came from a goldfish or a piece of dirt you found on, Atwater Avenue, but it's a chemical, and you find it increases the activity of the protein whose activity you want to increase. But then you have to say, okay, now that's in a test tube or on a dish. How do I take that to an animal? So you have to make sure that it works in the animal, that it doesn't make the animal sick. Then you take it to people, and after you do the right ethics review committees, and you start with very small doses of the chemical in people, and then you go to a large number of people at higher doses, and so on. And so there's a graduated phasing of things. And this is a risky business. You don't want the last thing you want to do is harm somebody.
No. Yes.
So that's one of the things. One of the other things, the benefits, though, I should refer to for these genetic diseases is gene therapy. for the 6,000 single gene diseases I mentioned, the simple idea is, well, if you're missing a gene, can you put a normal copy back in and correct the disease? And in concept, this is brilliantly simple, right? In execution, it's much more complicated. You have to get it into the right organ. So in the case of the eye is accessible. And so there are now 3 human eye diseases, genetic ones, where in animal models, you can put the normal gene back in and prevent the visual loss, or actually restore vision. And so, and there's about 5 immune system diseases where gene therapy has also been successful. You're essentially curing the disease. This won't be possible all the time. The eye works because you can get at it. And the immune system disease works because they involve bone marrow and cells you can take out of the blood and put back in. If you've got, part of the brain affected, the challenge is different and more difficult. Although there's pretty exciting advances occurring in that area too. So.
And with all of this research going on, you're talking about your research and some that you're aware of, but it's a big planet and there are a lot of countries that are involved in this. And so we're just wondering, how do you avoid duplication of research and research effort, or is the overlap and the competition actually productive?
Yeah, that's a good question. You can't avoid it. So how do you avoid the competition? It's the competition that keeps you awake at night and makes you get up early in the morning. You try to be aware of what other people in your area are doing, and if they're decent people, which most of the time they aren't, but not always, you tell them what you're doing so that you and they don't do the same thing. It's kind of a waste of time. Often if 2 of you are approaching the same problem from somewhat different directions, the outcome is what you referred to, is that the results are complementary rather than in conflict. But the number of instances in which you get scooped, which is what you're referring to, is decreasing because the number of problems that are studyable is so vast that the chance of somebody studying the exact same thing as you is less than it used to be previously. With 23,000 genes to study, and asking different questions about those genes, there's a lot out there to study. It's much easier now to ask a really good research question, which might be, what does this gene that I referred to earlier, what does that do, than it was when I started in science, where coming up with a really good question was actually much more difficult. So now you don't have to be, you can be a great scientist without being particularly imaginative because the question is there already for you. You just, if you're a great problem solver, you can do famously well.
And from our discussion this evening, one senses a tremendous flow of information. How is it communicated within the realm of research? You were saying something earlier about your this David Ng and how he went looking. So how are researchers aware of what's out there and what's being done?
Through the published literature. I mean, so all the journals now where we publish, there's a vast number of journals that we all use. They all have online versions, and so and the Library of Medicine and the National Institutes of Health in the U.S. is magnificent in this regard. Every journal, every paper in every journal is available, sometimes at a cost, because some of them are owned privately, but is accessible. And so the university like McGill will buy access to the whole literature. I can, and then I can tell the computer at the National Library of Medicine in the U.S. to send me every paper published, every new paper on retinal degeneration. It'll just automatically feed it to me. Or to my lab, and so it's a deluge, and so then you restrict it more and more. But that's you can't possibly keep up on everything, so you try to focus it and be narrow, but still studying something important. Without computers have made this.
Yes, it's interesting, isn't it? The genome and the computer, they seem to be complementary. They facilitate this tremendous flood of information.
Exactly. Each genome has 3 billion bits of information.
We're talking an awful lot about genes, but I'm trying to visualize what a gene actually looks like. Can we just get
It's a sequence of letters.
Okay.
A, C, T, or G, an endless number of combinations. So if we went to a
And then when all
computer,
the genes are fitted together, you get different combinations.
That's
as
exactly
well.
right, yeah. So it's an endless series of variations of the 4 letters A, C, T, and G, which refer to the chemical components of the gene. Yeah.
Well, I suppose we're lucky we only have 4 letters.
Well, computers are made for this kind of stuff. So.
Yes. Okay. There was something in your text which we puzzled over, and it was the phrase personalized medicine. And we wondered whether this meant that somehow the doctor-patient relationship would be much deeper or richer. But I don't think that's what you meant.
No, it isn't what it meant. It meant that personalized medicine is a term which recognizes that people who appear on a medical level, even with medical tests and the doctor's clinical exam and lab tests, to have exactly the same disease. When you go look at the cells affected, you realize the 2 people who seem the same don't have exactly the same disease. So let me give you an example. So acute leukemias, okay?
Yes.
2 patients in the hospital with seemingly identical diseases, which you would call acute leukemia. When you look at the DNA abnormalities in the leukemic cells, you realize they're not the same at all. I mean, there's more similarities between them than between that and a breast cancer genome. And so the importance of this is that if the 2 patients with so-called acute leukemia don't have exactly the same changes in the cancer is a genetic disease, okay? It's not inherited most of the time, it's acquired mutations. So if patient A and patient B don't have exactly the same disturbances in their leukemic cells, you can imagine, well, maybe the treatment and the prognosis are different, and that's absolutely the case. So you now take patients with many of these cancers. The front line in personalized medicine is cancer. And so the recognition that everybody's cancer cells are different. Okay, there's no 2 humans on Earth who have breast cancer who have exactly the same abnormalities, for example. And so you take patient A with acute leukemia and patient B, you study their genome and realize, okay, we've seen 100 patients with changes like patient A before, and they respond well to drug number 1. Right. And patients like who have changes like the other patient, patient B, respond better to drug 3, and in fact might have adverse responses to drug 1. So the personalization is in the kind of prognosis and classification and treatment you give. So this is transforming cancer therapy.
Perhaps we can actually look at those 2 areas, cancer and Alzheimer's. What are the immediate prospects? I mean, for example, I heard the other day on the news of a case of a man who had been diagnosed with leukemia. He was given a particular drug, and within a week, the problem had been treated, cured. Well, he was in remission, I guess. Are these advances truly possible, or was this just...
No, that happens. I mean, he's a lucky fellow.
Yeah, very. Right.
Let's hope that it doesn't occur.
Right.
Yeah. Cancer is a moving target, unfortunately, because the cancer cell abnormality isn't static. The cancer keeps on evolving. And cancer is actually just genetic chaos, that's what it is. And so the genetic chaos, this cancer cell is always changing, and so the reason a cancer cell becomes resistant to a drug that the patient initially responded to is because the cancer DNA has changed yet again, and the effect of the drug is no longer there. And so that's why it's a moving target. And so it's tough. It's tough to treat.
It's not really related to one particular organ, and it can spread to other organs as well.
Yeah, but that's true. But even if you stick with this, if you stick with blood cancers, for example, white blood cell cancers, you have to find a drug that will treat and stop the cancer dead every time is a magnificent achievement. It's trial and error. Okay, and so there are some cancers which are fabulously treatable. The treatment having been developed over decades of trial and error.